Good evening, ladies and gentlemen. I'm a graduate student of Fudan University. I feel honored to attend this meeting. Next, I will introduce the effect of 919 syrup on regulation of the Ghrelin pathway and the intestinal microbiota in rats with non-alcoholic fatty liver disease. First of all, I will introduce the experimental design. It has two parts. Timeline of 919 syrup's toxicity test. 20 male SD rats were randomly divided into the control and the 919 TT groups. Both groups of rats were fed continuously for 4 weeks, and daily body weight and feed intake were recorded. At the beginning of the fifth week, the rats were sacrificed, blood and liver samples collected, serum biochemical index, blood glucose, and hepatorenal function were detected, and liver histological changes were observed by HE staining. Timeline of experimental assessing effect of 919 syrup on NAFLD rats. Third, male SD rats were randomly divided into control NAFLD and 919 TT groups. The control group received the standard diet, while the NAFLD and 919 TT groups were fed the high-fat diet for 9 weeks to establish NAFLD. Liver HE staining confirmed that disease were established. From the beginning of the 10th week, rats in the 919 TT group were given 919 syrup by gavage. This method of feeding continued for 4 weeks, and body weight and feed intake were recorded daily. At the beginning of week 14, fecal samples were collected. Hypothalamus, stomach, and liver samples were collected to detect the expression of genes and proteins associated with the brainstorm Ghrelin pathway. Next, toxicity test of the 919 syrup. This picture shows changes in body weight and feed intake in the two groups of rats over 4 weeks. After 4 weeks, there were no significant difference in body weight and feed intake between the control and 919 TT groups. These results show that 919 syrup did not negatively impact the body weight and feed intake of normal rats. The first picture shows blood glucose levels in two groups of rats after 4 weeks of feeding, and there was no statistical difference between the two. These results show that 919 syrup didn't negatively impact the blood glucose of normal rats. The second picture showed changes in hepatorenal function indicators AL2-CR after 4 weeks of feeding, and there were no statistical difference between the two groups. These results show that 919 syrup has no obviously negative impact on the hepatorenal function of normal rats. This picture shows HE staining images of rats' livers in both groups after 4 weeks of feeding. HE images of the rats' livers from both groups show that liver cells were normal in shape, orderly in arrangement, and readily arranged from the center of the central one. These results show that 919 syrup does not negatively impact the liver tissue structure of normal rats. Therefore, we can come to a conclusion that Reasonable doses of 919 syrup has no toxicity on rats. Next, we'll talk about the effect of 919 syrup on the peptide-related Ghrelin pathway in non-alcoholic fatty liver disease rats. From the beginning of the 10th week, rats in the 9-1 group were given 919 syrup by gavage, and the rats in the NAFO group were given the same volume of normal saline by gavage. Body weight and feed intake were recorded during gavage, as shown in the picture. Compared with the rats in the NAFO group, the weight and feed intake of the rats in the 919 group decreased after feeding with 919 syrup during the gavage period. This picture shows changes in expression of Ghrelin pathway-related genes in the hypothalamus and stomach of the three groups of rats. As compared to the control group, 582CR, 582BR, as leptin, AGRP, NPY, and CRF expression increased in the hypothalamus and stomach of NAFLD rats, while Ghrelin and GHSR expression decreased in both sides. As compared to the NAFLD group, 582CR, 582BR, leptin, AGRP, NPY, and CRF expression decreased in the hypothalamus and stomach of the 919TJ group, while Ghrelin and GHSR expression increased in both sides. This result shows that 919 syrup can reverse the abnormal expression of Ghrelin pathway-related genes in the hypothalamus and stomach of NAFL rats. Then we detect Ghrelin protein levels in the liver of rats in the three groups. As compared to the control group, Ghrelin protein levels decreased in the liver of rats in the NAFL groups. As compared to the NAFL group, Ghrelin protein levels increased in the liver of rats in the 919TJ group. Results show that 919 syrup can recover Ghrelin protein levels in the liver of rats with NAFL. Next, we will talk about the effect of 919 syrup on intestinal microbiota in rats with non-alcoholic fatty liver disease. First, we'll talk about correlation analysis of intestinal dominant microbiota and Ghrelin pathway. Experiment correlation analysis was used to explore the relationship between intestinal microbiota and the Ghrelin pathway in rats. Correlation analysis between the three groups of rats dominant intestinal microbiota and Ghrelin pathway-related molecules were performed, and the heat maps were used to represent the results. The left picture shows the correlation between the dominant intestinal strains and the phylum levels and molecules in the hypothalamic Ghrelin pathway. Tenericates had a significant positive correlation with POMC, CRF, NPY, 582CR, leptin, AGRP, and 582BR. Spiral caches have a significant positive correlation with AGRP and 582CR. Cyanobacteria had a significant positive correlation with Ghrelin, and Wurmtag bacteria had a significant negative correlation with POMC and Ghrelin. The right picture shows the correlation between the dominant intestinal strains and the genus level and the molecules in the hypothalamic Ghrelin pathway. Chapnoma 2 had a significant positive correlation with AGRP and 5-2BR. Viscera has a significant positive correlation with CRF-MPY, POMC, and Ghrelin. And no-rank have numerous bacteria and rhomboidea were negatively correlated with CHSR. Next, the left picture shows the correlation between the dominant intestinal strains and the phylum level and gastric Ghrelin pathway specific molecules. PDC bacteria was negatively correlated with GHSR. Spirochetes had a significant positive correlation with AGRP, and bacteria had a significant negative correlation with AGRP. The right picture shows the correlation between dominant intestinal strains and the genus level and the molecules in the Ghrelin pathway in the stomach. Viscera had a significant positive correlation with GHSR, POMC, and Ghrelin. Rhomboidea and Trampoma 2 had a significant positive correlation with HRP, and no-rank F bacteria had a significant positive correlation with MPY. These results reveal the dominant microbiota in connection with Ghrelin pathway and indicate that this bacteria may be related to nephod. In brief, this study showed that 919 syrup had non-negative effects on body weight, feed intake, blood glucose, hepatorenal function, and liver histology or normalized. 919 syrup reversed the abnormal gene expression of the Ghrelin pathway in brain and stomach and nephod conditions, regulated feed intake and body weight, and improved intestinal microbiota disturbance in US with nephod. In summary, 919 syrup have therapeutic effects on nephod by regulating feed intake, body weight, and the balance of intestinal microbiota, and is a promising therapeutic drug for treatment of nephod. Okay, that's all. Thank you.